In the biomolecules chapter, we must have discussed something about this, saying that in nature there are amino acids of more than 180 available, that is present in the cell. But all of these 180, they are not associated with the formation of proteins. In fact, there are 20, and in the recent studies, it is 21 amino acids only that are associated with the formation of proteins. The rest of the amino acids, they act as activators, they act as cofactors for many biological processes, okay, within the cell or within a system. So there are 20 which are involved in this, and these 20 are supposed to be coded by a total of 4 nitrogen bases, because on the DNA you have only 4 nitrogen bases: adenine, thymine, guanine and cytosine. And we say that which amino acid should come at which position. The code for that particular amino acid is present on the DNA. If I have a DNA like this, and if it has nucleotides in this position, let us say in the same ATGC format, it is said that these will be coding for the amino acids, the order of the amino acids and the sequence of amino acids, as we have already discussed in the introduction of transcription. Now here the number is found to be a little weird. How is it possible that you have 20 amino acids that can be coded by only 4 nitrogen bases? It's a little, the probability is found to be a little weird, right? And there were many theories behind it, but the most accepted theory was given by Francois Crick, and according to this theory, he suggested the existence of a genetic code. Francis Crick or Francois Crick, he suggested the existence of a genetic code, where he said that it is this smallest code. There is the genetic code has a smallest unit, like taxonomy has a smallest unit species, right? Similarly, genetic code also has a smallest unit, and each smallest unit of a genetic code will code for one amino acid. Okay, and the name for this smallest unit is called codon. Okay, so each codon codes for one amino acid. Clear? Okay. So genetic code basically is defined as a code made up of codons. It is a group of codons which will code for a particular, and in that each codon will be coding for an amino acid. Okay, fine, that is clear. Now, how many nucleotides make up the codon? How many nucleotides make up the codon? Is it one nucleotide, two nucleotides, three, four? So, how many nucleotides are present in the codon? Now, the answer to this question was given by a mathematician called 𝐄𝐻𝐫. So, 𝐄𝐻𝐫 was a mathematician and he gave an answer. So what he has done is he has done simple probabilities. He has created simple probabilities to understand it. So there are 4 nitrogen bases. What we know is 4 nitrogen bases and 20 amino acids. Okay. First, what Gamow said is, let us take that each codon consists of one nucleotide. Then 4 nucleotides code for only 4 amino acids. Correct? So this is completely insufficient, because the number that we want, the number we are looking for is 20 amino acids. So this is insufficient. Next, he went to the second section. He said that what if each codon consists of 2 nucleotides, then it will be again 4 to the power of 2. Correct? What it will be? 4 into 4, that is 16 amino acids. Yes, almost are there, but it is not still complete. Even if you are having 16, it is still insufficient because for 20 amino acids. So again we say it is insufficient. Then he said, what about the case where my codon contains 3 nucleotides? That means 4 to the power of 3, which is 4 into 4 into 4. That means 64. Now here you say, oh my God, ma'am, it is way too much than the number we require. But if you think about it logically, 1 and 2 conditions, that is 1 nucleotide and 2 nucleotides, are insufficient. 64 is over sufficient. 3 nucleotides is over sufficient. So there's no point going to 4 nucleotides because that will be even more. But what if even though it is more than enough, more than required is there, correct, you are true. What if several of the triplets have the same letters but in different sequence? Okay, he said that in the 64 you might have the same letters but different sequence. Means he is saying ATG can also be AGT. Correct? It can also be TAG, it can also be GAT, and another combination is it can be TGA and GTA. So the number of letters are the same. You have the same letters A, T, G, but it is present in different combinations. Correct? So if you are getting these different combinations, then it is possible that you will have 64 combinations. Correct? So, and it is possible that these code for different amino acids. Whatever amino acid ATG is coding, it is not necessary that AGT will code for it, TAG will also code for it, or GAT might code the same amino acid as ATG. So they might code for different amino acids. That is a probability, right? So why not 64 codons? Okay, so this is what he has said, the Gh's version. And according to Gh's, he said that a single codon is triplet in nature. It's a triplet codon, means it is made up of 3 nucleotides. But again, he is a mathematician trying to prove a biological phenomena. Obviously everybody will have a lot of doubts the same way Mendel had, because Mendel tried to explain a biological phenomena in the statistical point of view, right? So later Gh's work was proven by 2 other scientists. It was one Nirenberg and Mathai, and another one is an Indian based scientist called Dr. 𝐵𝐺𝐵 They have come up with proofs that showed that the codon is triplet in nature. Okay, so let us see what they said. So it was Dr. 𝐵𝐺𝐵, Dr. 𝐵𝐺𝐵, and along with Nirenberg, because Nirenberg provided what is called as a cell-free translation system. That is, you can translate an mRNA sequence without a cell. And for that, they have used specifically an enzyme called as the 𝐶𝑠 polymerase. 𝐶𝑠 polymerase. It is an enzyme that is useful when carrying out the translation in a cell-free system. So what they have done? First, they have synthesized an RNA which is made up of same nucleotides, which is called as a homopolymer. Okay, now they knew how many nucleotides they were adding. Let us say in the first case it is poly-U. So that means you're taking multiple U's. 1, 2, 3, 4, 5, 6, 7, 8 and 9. So this is a poly-U chain. When they put this poly-U chain in a cell-free translation system, they have got everything as a long chain of phenylalanine. So the only single amino acid they have found, and the polypeptide, all the amino acids were phenylalanine in nature. And how many phenylalanine? They've got a very particular specific number of phenylalanine as 3. I mean, I'm hypothetically saying 3, they did not take 3. So they got 3 phenylalanine amino acids in that chain. If you look here, how many, if you say the codon is triplet in nature, then these 3 use code for 1 amino acid, these 3 code for the second amino acid, and these 3 code for the third amino acid. So you will get 3 amino acids, and because everything is same, it should be phenylalanine, hence you are getting 3 phenylalanine. Correct? If it's a doublet, that means if it's 2 nucleotides, then how many you should get? 1, 2, 3, 4. You should get 4 phenylalanine. You did not get that. So obviously the codon is triplet in nature. He did not stop there. He used again poly C. He used poly C. Okay, poly C, and this time poly C is giving a completely different, it is giving a completely different amino acid, which is proline. Okay, and how many proline? 1, 2, that's it. That means what they have done is they have counted the number of bases they are keeping, and they are counting the number of amino acids they got. So based on the number of bases present in their homopolymer and the number of amino acids they got in the polypeptide chain, they were able to conclude that it was a triplet codon. Okay, then they started to do something called as copolymers. Now what is this copolymer? Now in copolymers, what they have done, they have taken an RNA polymer which is made up of multiple different bases like UUCCCA, and let me add another one, CAA, okay, another one ACA, like this. So totally how many codons will be present now if I have to give it? 1, 2, 3, sorry, and 4. So 4 codons, that should give 4 amino acids. Now it was found that UUC was giving phenylalanine. This was giving phenylalanine. Okay, then CCA was giving proline. It was giving proline. Then CAA, it was giving glutamic acid. Okay, and ACA, sorry for that, ACA was giving an amino acid called threonine. If you now look very carefully, CCA was giving proline, and they also found that CCC was also giving proline. So with this, they found that it is not just one codon coding for one specific amino acids. They said that 2 different codons can code for the same amino acid. You have 2 different combinations of 2 different codons, but both of them are coding for the same amino acid. Correct? Similarly is the case of this one, UUU and UUC, again phenylalanine. So if this is the case, then that 64 codons can be explained, right? You might have multiple codons coding for the same amino acid. In that way, the 20 amino acids, 64 codons is easier to understand. Right. So this phenomena later became or is called later as degeneracy. So like this, later on an entire genetic code was established by like this in the same way by identifying which combination is coding for which amino acid. So this is the genetic code. And now if we look at this genetic code in a little detailed manner, we will see that out of these 64, this includes all the 64 codons. Right. Now out of these 64, if you see specifically there are 3 codons here, another one here, and another one here. Okay, so I'm specifically having 3 codons like this, which are saying stop codons. They're particularly using the word stop codons. So UAA, UAG and UGA. These 3 codons are called stop codons. Now what do you mean by stop codons? They terminate or stop translation process. Just like you have a terminator for transcription, these codons stop the translation process. So that means if I make a math, out of 64 codons, 3 are stop codons. So the ones that are actually coding for the 61, you have only 61 codons that are actually coding for the 20 amino acids. So again, there is a little less in the number. Now, then if you see another specific codon, an interesting codon here, this fellow. Okay, this AUG. If you look at it, AUG has 2 roles. Okay, start plus met. So this is called as a dual codon. Means this one codon has 2 functions. One, it will code for methionine. The first function is it is coding for methionine, and second function is it is acting as a initiator codon. Initiator for what? Initiator for the process of translation. So when I talk about the salient features of genetic code, the first code is that codon is triplet. Okay, so codon is a triplet codon. In this triplet codon, 61 code for amino acids and 3 are stop codons. This is the second point. The third point is the codon AUG has dual function where it codes for methionine and it also acts as an initiator. Okay, then if you look at the entire genetic code, you will find that each codon will code for only one amino acid. For example, if you take the case of GCU, it will code only for alanine. You will not find GCU coding for any other amino acid also like valine or asparagine or serine. GCU is associated with only alanine. Similarly, UUU is associated with only phenylalanine. U will not be coding any other amino acid like let us say lysine or alanine. So each codon is specific for only one amino acid. So this nature is called specificity. Each amino acid, one codon codes for only one amino acid and therefore it is unambiguous. There will be no confusion at all. And if you see, now let's go to this area now. If you see here, I have 4 different codons. All of them are coding for the same amino acid. Okay. Similarly, I have 2 different codons coding for the same amino acid. So like I have said just now, one codon will code for one amino acid, but different codons can code for the same amino acid, and this nature is called degeneracy. Okay, so this nature is called degeneracy. So degenerate is when some amino acids are coded by more than one codon. So that will be the 4th point. And finally, the 5th one is this genetic code is universal. Whether it is bacteria, whether it is human being, whether it is a plant. Okay, UCU will always code for serine. It is fixed. There will be no change. The only exception is the mitochondrial genetic code. Remember when we were talking about cell, the unit of life, we said that mitochondria has its own genome, right? Now because mitochondria has its own genome, there it will have its own genetic code. That will be exception. But nuclear genome's genetic code, no matter whether you're a bacteria, you're a fungi, you're an algae, you're a human being, you're a platypus, you're a kangaroo, no matter what you are, your genetic code is universally the same. Okay, so this is your genetic code. Normally here we discuss about verbal hypothesis, but on the back of my mind, on a hindsight, I would like to discuss it after we discuss the next structure, that is tRNA, because when we are discussing verbal hypothesis, we are going to use the term anticodon. To understand anticodon, it's better you have a clarity about the tRNA structure, because anticodons are present on the tRNA. So at tRNA, I will talk about the verbal hypothesis, which very clearly explains this degeneracy phenomena that you are seeing in the genetic code. I hope this topic is clear to all of you.